Welcome, and thank you for joining us today for the webinar, Intermittent Fasting and Time-Restricted Eating, Implications for Biomarkers, Body Composition, and Cancer Outcomes. My name is Stephanie, and I will be your WebEx host. Before we move into introducing our speakers for today, I'd like to make a few comments before we get started. All lines have been muted upon entry and will remain muted for the duration of the webinar. Please submit your questions throughout the presentation in the Q&A or chat panel and select all panelists from the drop-down. We will ask these questions on your behalf during the Q&A portion of the webinar. If you need to view live captioning, please refer to the Media Viewer panel. This webinar is being recorded and it will be posted online in the near future. And now I'd like to turn it over to our moderators, Drs. Boyd, O'Connor, and Sauter.
Hi, my name is Pat Boyd, and I'm joined by Dr. Sydney O'Connor and Ed Sauter, who will be also moderating this webinar panel here at the National Cancer Institute. Recently at NCI, we've been examining the state of research in the areas of intermittent fasting and time-restricted eating. Ed Sauter here, one of our moderators, has spearheaded many of these efforts, including being the lead on one of NCI's 2020 provocative questions, and specifically provocative question number 2, which relates to intermittent fasting. This provocative question asks how intermittent fasting affects cancer incidence, treatment responses, or outcomes, and the website link to that PQ is provided on the next slide. And Dr. Sauter will touch more upon provocative question number 2 after the panelists all present. As many joining probably know, there's been a growing interest in intermittent fasting and time-restricted eating over the last 4 years or so. If you take a look at Google searches on intermittent fasting and time-restricted feeding, those have grown dramatically since 2016, as well as if you search for these terms on PubMed as well, you see a steady growth over the last 4 years or so. So of course, because of this growing interest, we're excited about our expert group of researchers to present on intermittent fasting and time-restricted feeding today. I'll hand it over to Dr. O'Connor to now introduce the team.
So today we're joined by 3 experts in the field of intermittent fasting and time-restricted eating. Our first speaker will be Dr. Sachin Panda from the Salk Institute in La Jolla. Our second speaker will be Dr. Dorothy Sears from ASU and UCSG. And our third speaker, Dr. Courtney Peterson, is from the University of Alabama, Birmingham. We've invited these 3 individuals because of their complementary backgrounds, areas of expertise, and approaches. So I'll go ahead and introduce our first speaker now, Dr. Panda. Dr. Sachin Panda is a full professor in the Regulatory Biology Department at the Salk Institute for Biological Studies. He received his PhD from the Scripps Research Institute in La Jolla and completed his postdoctoral work at the Genomics Institute of the Novartis Research Foundation in San Diego, California. Dr. Panda's group is interested in understanding the molecular mechanisms of the biological clock and, among other things, the potential benefits of time-restricted feeding under different conditions of sleep activity and disease. Welcome, Dr. Panda.
Hi. Okay, I guess you can hear me. What I'll do is I'll give you the concept of time-restricted feeding or eating and how this can be used in practice, what are the current hurdles and what can be done. See, that's down. Okay, so the word time-restricted feeding or time-restricted eating was practically coined in our lab in 2012, and now it's synonymously used in popular media with intermittent fasting. But if you do PubMed searches, then there are some key differences, so that's why I wanted to have this slide. So intermittent fasting has its root in the concept of reducing total calorie intake for 1 or more days in a week or month. And as caloric restriction improves health, this form of intermittent fasting can improve health in both animal models and humans. And most of the intermittent fasting studies involved alternate day fasting or reducing daily caloric intake or 2 or 3 days of fasting does not take into account the timing of food intake. Whereas time-restricted feeding, that is term used to describe animal studies, or time-restricted eating used to describe human studies, has its root in circadian rhythms, and it implies that restricting the number of hours of caloric intake to a consistent window of 12 hours or less daily without an overt attempt at reducing calorie can improve health. So that's the key difference between these two terms. And why this is important is particularly for researchers who are trying to get some literature on this time restricting or intermittent fasting, they should be mindful about this difference. Okay, so let's see. So with the time restricted eating, it was for a long time it was known that reducing caloric intake or the number of calories does matter. Reducing caloric intake can improve health. And we also knew that nutrition quality matters. So macronutrient imbalance can also put somebody at high risk for various diseases. And the time-restricted eating literature essentially added another leg to nutrition science, that is the timing of calorie also matters. So in this way, the quality, quantity, and timing of caloric intake do matter when it comes to health. Over the last 8 years, starting from 2012, there are many preclinical animal models of time-restricted feeding studies. And those studies have shown that in mouse model or in Drosophila fly model, time-restricted feeding can improve quite a few health parameters, and many of them are also related to cancer risk. So now the question is, how do we go from animal studies to human studies? At the same time, use animal studies to tease apart mechanism, because the human studies may not be sufficient enough to tease apart mechanisms. And in this context, the pathway to practice is kind of well laid out for the field. The animal studies started from 2012 onwards, and the pilot feasibility studies in humans started from 2015 onwards. And in the last 2 to 3 years, now we are seeing pilot intervention studies in patients who have some metabolic disease. And then hopefully in the next few years, we'll see this path to practice will go on. At the same time, the animal studies will also continue to understand mechanisms. So now some background on time-restricted feeding studies, what have been published, because when we say PRF, it can be feeding anywhere from say 4 hours to 12 hours. And then the question is, what time of the day or night mice were fed, should be fed, and what kind of outcomes we would expect. Whether the studies are done in young or old mice, which strain, male or female, all of these have to be kept in mind to give an idea about what is missing, what is the knowledge gap, and where we should be doing more research. So the initial study on time-restricted feeding were actually published from my lab, that's the paper, and also simultaneously by Oren Froy's lab from Israel, came out around the same time in the same week. So our study used high-fat diet, 60% calories from fat, and also normal diet, the standard diet that is used in most vivariums. These mice were B6 male mice, and they were young, starting from 12 weeks of age. Those mice got 8 hours of food during the dark phase, when mice usually eat. And this was followed up by another study from my lab in 2014, because the first study showed prevention of obesity and many metabolic disease, but not treatment or reversal. So that means we showed that the normal mice, when they are on time-restricted feeding, they are protected from obesity and metabolic disease. So in the second paper from our lab, Amandine took 390 plus mice, put them on high-fat diet, high-fat, high-sucrose diet, and normal diet. They were again young male B6 mice. They got 9 hours of time-restricted feeding, 12 hours of time-restricted feeding at night, of 15 hours of time-restricted feeding that spanned late afternoon to morning. And then she also did another interesting study where for 5 days, these mice were on 9 hours of time-restricted feeding, and for 2 days in the weekend, they got ad-lib feeding. And what we found is the ones that are in green, we found protection from obesity and many metabolic diseases. But when mice had for 15 hours or longer, then that protection was not that great, or it actually didn't stay for longer. Amandine also did a treatment approach where she put, again, young or middle-aged B6 mice. Those were already on high-fat diet ad libitum. They were already unhealthy, and put them on 9 hours of time-restricted feeding. And to our surprise, time-restricted feeding could also act as a treatment. So on pre-existing obesity and metabolic disease, TRF had benefit. Then subsequently, Amir Jarinpar from our lab again did another time-restricted feeding study with 8 hours time-restricted feeding at night to show that time-restricted feeding does change certain aspects of microbiome. This is a little bit more complicated. We don't have time for that, but you can take the references. And then there was this idea that circadian clock may be necessary to mediate the effect of time-restricted feeding. But at the same time, we saw many nutrient sensing pathways are modulated by time-restricted feeding. So Amandine put young circadian mutant mice, most of them are males, on high-fat, high-sucrose diet. And when there are 9 hours time-restricted feeding, that could still give many benefits of time-restricted feeding. And this was exciting for us because it showed that the circadian mutant mice were predisposed to metabolic disease. They can also be treated, or time-restricted feeding can prevent obesity in that. This is exciting in the sense that many people think that there is a genetic basis for their obesity and metabolic disease. And the question now is, can good behavior dampen the bad effects of bad genes? So this was the beginning of those studies. As I said, most of these studies, almost all of these studies from our lab so far have been on young male mice. And there is another study here that actually used female mice, overectomized female mice, and it was a treatment model too, and essentially time-restricted feeding on overectomized female mice, which mimics postmenopausal women's health, was also effective. And as I mentioned, there was one study from Israel that showed 4 hours of time-restricted feeding, and in this case, this 4 hours was during daytime, not at nighttime, was still effective in preventing metabolic disease. So many people think that daytime feeding is bad for mice, but actually daytime time-restricted feeding may be better than ad libitum feeding. In fact, there are a few other studies. So for example, Arbol et al. Showed that daytime feeding was, when mice were fed during daytime, they weighed slightly more than mice fed during nighttime. That led the idea that circadian misalignment feeding is bad for us. But I must stress that this study was a little underpowered. There were only 6 mice per group, and there was no control group to show whether these daytime fed mice, in fact, weighed less than ad lib fed mice. And other groups have showed that, yes, even daytime feeding, time-restricted feeding gives many, not all benefits. So it's better to put your mice on daytime feeding than ad libitum feeding. But at the same time, there's also confounding aspect of sleep disruption, because mice usually sleep during daytime, and when you feed them during daytime, you're disrupting their sleep, because they cannot fully recover. So the bad effect of daytime feeding in mice cannot be ascribed to daytime feeding only, but to sleep disruption. So in light of this, Joe Takahashi has gone back and checked the impact of traditional caloric restriction studies. Most CR studies are done in a way where the mice are fed 70% of their daily calories at the beginning of the night or beginning of the day. So he compared this 12 hours day TRF, 12 hours night TRF, daytime caloric restriction, nighttime caloric restriction. Although there are small differences among these those two, the bottom line is any one of these interventions was better than ad libitum feeding. So we have to keep that in mind, that time restriction is better than no restriction, and certain aspect of caloric restriction, as described in mouse literature at least, also has a time restriction aspect. Then what are the benefits of time-restricted feeding? These are some of this is the summary of many of the mouse studies, showing that when mice eat ad libitum, particularly a energy-dense diet, their circadian rhythms have low amplitude, so they are not that strong. And under time-restricted feeding, the circadian clock components or their output genes or the outputs that are influenced by nutrient sensing mechanism show very strong up and down or high amplitude rhythms. And that ultimately lead to, we don't know how much causal relationship is there, there may be some, to reduced adiposity, improved glucose tolerance, leptin resistance, liver pathology, all of these things are improved. Then the question is, what is the connection between circadian rhythm or time-restricted feeding to hallmarks of cancer? And can this be used to prevent, manage, or even rehab cancer patients? People are familiar with the cancer hallmarks, and what is interesting is many aspects of cancer hallmarks have a circadian component. For example, autophagy, metabolism, proliferation, oxidative stress, DNA damage response, immune surveillance, inflammation. These have circadian aspect to them, and we know that in many cancer types, the circadian aspect is broken. So that leads to the idea that maybe preserving robust circadian rhythm can impact some of the hallmarks of cancer, and that may be the potential pathway to use time-restricted feeding or any kind of interventions that improve circadian rhythm to reduce the burden of cancer. And so the concept of, because most people think that light is the measure and training cue for circadian rhythm, but over the last several years, studies from multiple labs are showing that timing of food intake is the dominant and training signal for circadian clocks in most organs and brain regions outside the suprachiasmatic nucleus. So in that way, TRF can sustain circadian rhythm, which in turn can reduce the risk of cancer by acting on molecular pathways such as mTOR and AMPK, autophagy, inflammatory cytokines, etc. Many of these have already been shown in quite a few papers. Organelle systems, so for example, ER stress and mitochondrial health can be improved by time-restricted feeding, and physiological states, obesity, glucose regulation, liver health, gut health, etc. However, there is a knowledge gap, that is an in-depth assessment of TRF on molecular changes at DNA methylation, chromatin modification, gene expression. All these molecular changes are currently lacking. So that's why we cannot put a finger on what is the mechanism of time-restricted feeding and which mechanisms may lead to reduced cancer burden. At the same time, we have to be mindful of some of the behavioral interventions as opposed to pharmacological interventions. Pharmacological interventions have defined doses, length of treatment, mode of administration, and outcome. The dose and methods used in clinical trials are translated to practice, whereas behavior interventions are often modified by individual researchers based on their own expertise, limitations, and academic interests. So the mode of delivery and length of treatment vary widely. And then the academic curiosity about quality, quantity, timing of nutrition, activity, sleep, mood, physiological states, and urge to test mechanisms often confuse and confound outcomes. There are more number of variables in a behavioral study than in a drug study, and that can be very challenging. So now the question is, we have been doing time-restricted feeding and some human studies on time-restricted eating, and what we feel, this is just a personal opinion so far, it has to be discussed and debated. We think that there has to be some reporting requirement in many of these time-restricted eating studies, at least in humans, So reference circadian timing, people always report their local timing, but however, humans don't live in under natural light-dark cycle, not even in they don't live in a defined light-dark cycle. So what should be the circadian time? Then sleep disruption and eating close to bedtime are known to affect metabolic homeostasis, so sleep should be reported. Then the biggest question is, what is a meal? Should coffee, beverages, small snacks be counted or should be reported? Then what is eating pattern? When it comes to eating pattern, it's really hard to define it. How many number of events and number of meals in a day, gap between meals, first caloric event, last caloric event, and then even macronutrient content of each meal. For example, having more carb in the morning or less carb in the morning is better, good or bad, we still have to figure out. Then cumulative caloric intake, because if we eat most of our calories by end of the day, even a small amount of calorie at night, is it more harmful? Then how many days of eating data we should collect, because people change their eating pattern during weekdays and weekends, and those things have to be taken into account. Then we have to measure activity and self-reporting. So what I'll do is I'll quickly go through what we do. We use an app called My Circadian Clock app that we developed in the lab. We use a continuous glucose monitor to monitor physiological response to feeding, and then we put an active watch. And I'll not go through this app, but it's a non-commercial HIPAA-compliant app. We are now running multiple studies. And what we do is our standard time-restricted eating intervention is to do 10 hours of self-selected time-restricted eating with attention to sleep time. So what it means is we advise our patients to be in bed for 8 hours and then avoid food for 1 to 2 hours after waking up, and then do 10 hours of time-restricted eating and make sure that there is at least 2 to 3 hours of gap before going to bed. And then if they want to have coffee, we say, well, have your coffee maybe a little after lunchtime and no coffee after that. So there are quite a few studies that we have done and some are ongoing studies. I'll talk about only one study that we just published recently, in which there are 19 patients were in this study, and out of them 16 were on medication for various metabolic disease. Our interventions are usually 12 to 14 weeks, and it's a 10 hours time restricted eating. And as I said, we put an active watch to measure sleep and activity and continuous glucose monitor. What we found is by doing time restricted eating, they could reduce their eating pattern. They had a 95% eating interval at 15 hours. They reduced it to 11 hours. And I'll get to why that 95% eating interval is important. So these are the eating event of one person. Each dot here is 1 calorie containing meal. If I look at just the average time from the first meal to last meal, it's only less than 12 hours, and most of the days it's around that. But the thing is, since circadian system is an anticipatory system, it's anticipating when the meal is coming every day. What we do is we figure out the time interval in which 95% of the meals would occur in 2 weeks interval. That's the going to bed time, that's the wake up time. You can see there is a huge variability in the first calorie, huge variability in the last calorie, and after intervention, this variability reduces, and then the interval also reduces. So in this case, the daily average is 9 hours, the 95% interval is 10 hours. And that's how we calculate. I'll not go into the detail because of the time constraint, but I'll show you one interesting result. So this is a person who actually ate breakfast, had dinner before 7 o'clock, but had a very bad glucose regulation. This is glucose data from 14 days. He was technically diabetic because the HbA1c was 6.7. The only thing that he did was to delay the breakfast to 9 o'clock at the same dinner time, and he could reduce glucose to normal level without reducing too much his body weight, because his BMI went from 45 to 43.2. And why we are, and then in the same study, out of those 19 patients, we found quite a few benefits. For example, the cholesterol levels decreased, LDL cholesterol decreased, but the HDL did not increase. Blood sugar was better controlled, body fat was better controlled, body weight, blood pressure improved. And what we find is in time-restricted eating, people see the benefit within a week or two, and their sleep quality improves, their sense of energy improves, hunger at bedtime decreases, and acid reflux reduces. So these 4 factors act as a positive feedback loop to reinforce time-restricted eating on a consistent basis. So now the questions with regards to cancer is, can TRE reduce the risk of cancer that are prevalent among shift workers? Then can TRE be an add-on therapy for cancer treatments? Because the last paper was an add-on therapy for metabolic syndrome treatment. And then can TRE be part of care as cancer rehab or prehab program? And then on mechanism, how TRE affects hallmarks of cancer, and what are the short-term benefits of TRE, and can these benefits be leveraged in a behavioral intervention manner to sustain adherence to long-term? And with that, I'll stop and pass it on to Dr. Sears.
All right, before we jump into Dr. Sears' presentation, I'd like to thank Dr. Panda for that great talk, and remind all of our audience that if you do have any questions for any of the speakers, that you can use the chat function within WebEx, and we'll be asking these questions at the end. So now I'd like to introduce Dr. Dorothy Sears. Dr. Dorothy Sears is a professor of nutrition in the College of Health Solutions at Arizona State University and an adjunct professor of medicine and family medicine and public health at the University of California, San Diego. She received her PhD in molecular biology and genetics from the Johns Hopkins University School of Medicine, where she also completed her postdoctoral work. Dr. Sears has an established transdisciplinary program of research in cardiometabolic disease and cancer, which spans from the molecular to the population health level. She is actively researching the health benefits associated with nightly fasting and circadian alignment of food intake. Welcome, Dr. Sears.
Thank you. All right. So I don't see my slide yet.
If you just click on the presentation, there we go.
Perfect. Okay, there we go. Okay, so thank you. So we'll just jump right in. The title of my talk is Food Intake Timing and Fasting and Associations with Cancer. So my topics are simply just introducing obesity and insulin resistance and how those are relevant to cancer, and then food intake timing and fasting in relation to cancer, and then some conclusions and take-home messages. So as we know, there's a global epidemic of obesity across the world and in the United States. And what a lot of people don't know, but I'm sure is not unfamiliar to this crowd, is that obesity is associated with an increase in risk for cancer. And currently we know that this risk is increased for at least 13 different types of cancers. And depending on who you ask, the count is up to 16 in some cases, but along with some of the classical cardiometabolic diseases shown on this slide. And in addition, there's an increased risk for insulin resistance, which is shown at the bottom there. And in general, the more obese a person is, the more insulin resistant they are. Insulin resistance is typically silent for the most part, and it is rarely measured clinically. But it is very important for us to think about with respect to cancer. And I'm going to say a little bit more about that. In the context of obesity, there is a whole milieu of crosstalk between organs that are affected by obesity, with a highly integrated system of circulating hormones, cytokines, and nutrients that are affected by the obese state. And what I want you to notice is the 2 red arrows pointing out insulin resistance and hyperinsulinemia that result from that. And these are central mechanistic drivers of the cancer risk component of obesity. And other components are also important and relevant for time-restricted feeding and intermittent fasting, but one of the major emphases of our work and my talk today are going to be around insulin resistance and hyperinsulinemia. Okay, so what is insulin resistance? So insulin resistance is a state when the action of the hormone insulin is impaired. The activity of Insulin is required for proper nutrient storage. So when a person is insulin resistant, they have inefficient storage of nutrients, particularly of glucose, but other nutrients, lipids, and amino acids as well. And resulting from this, people have compensatory hyperinsulinemia. So this occurs both in the fasting state and the postprandial state. So this means after a meal. The postprandial means after a meal. And also the people with insulin resistance have impaired glycemic control or blood glucose control. And if the person doesn't have diabetes yet or is not pre-diabetic, they may have normal fasting levels of glucose, but they will have elevated postprandial glucose, meaning after a meal they're going to have very high exposure to glucose. And what happens is because of this very high level of blood glucose after meals, and like Dr. Panda was pointing out, we know people are eating all the time, so they're almost always, and many people are frequently or almost all the time in a postprandial state. Their proteins that are exposed to the blood are getting glycated or sugar-coated. And one of these that is used clinically to diagnose insulin or poor glycemic control or to diagnose diabetes is hemoglobin. So hemoglobin A1C is a glycated form of hemoglobin. And then another marker for insulin resistance is that it's associated with systemic inflammation, and clinically that's measured by C-reactive protein, although there are many other cytokines and inflammatory markers that are used in research. So the bottom line is that insulin resistance is a very strong mechanistic driver of cancer risk. I'm not showing all the data here, but it is it's huge. High glucose, insulin, and inflammation are all promoting tumor growth. Insulin resistance is always also quite prevalent worldwide. It affects 1/3 of the non-diabetic population in the U.S. It's almost 90 million people right now would be classified as having prediabetes. And again, when you add in the people with type 2 diabetes, then this number goes up, because when people have type 2 diabetes, they still have their insulin resistance. And we know that cancer risk is increased in people who have type 2 diabetes. Contributing factors are genetics. Can't do anything about that, but we can do something about the environmental risk factors, including our diet and our lifestyle behaviors, including the timing of when we eat. So interventions to reduce insulin resistance will have impacts on almost all the top causes of non-communicable, non-accidental deaths, including cancer. So we know that you can exercise and have diet, all these different diet plans for cancer risk reduction, including, moderate to vigorous physical activity, which is a guideline level of 115 minutes a week, calorie restriction, improving your diet quality, eating less fat or eating more fat if it's the keto, and there are all these diet plans. But the problems with these plans is that there are mixed results with these different approaches. They do work. Some of them have some insufficient data right now. Like I would say keto, we're still looking at that. But there's also poor compliance for some of these approaches, and they're not sustainable. So we are really interested in these time-restricted feeding or prolonged fasting or avoiding eating at night because we think that these might be more approachable, more sustainable approaches to cancer risk reduction at a population health level. So I'll get into that right now. Feeding behaviors can modify risk for diseases associated with insulin resistance and inflammation, including cancer. So intermittent fasting and health benefit, as Dr. Panda mentioned, there are many studies demonstrating improvements in weight and markers for chronic disease and cancer risk. The evidence is particularly strong in the mouse models and increasingly suggestive in humans. Currently, there are about 24 mostly underpowered clinical trials. So I'm very excited to see increasing support from NIH and NCI to fund more appropriately powered trials for this work in humans. Many of the intermittent fasting regimens, including skipping a day, not eating, or cutting down caloric intake tremendously throughout the waking period, I would not call these, and many others are not calling these, real-world feasible. Primarily for the second point, they're associated with hunger, mood changes because of this daytime fasting. In addition, as Dr. Panda alluded to, they're not aligned with the circadian rhythm light-dark cycle, and I'm going to get into more of that, about that. So this is some of the different intermittent fasting approaches that have been published. Again, you can see the sample number is quite small for these types of approaches, but I highlight the effects on insulin in these approaches. They indeed do tend to reduce insulin. Again, very small studies, but they're promising. But again, in the blue at the bottom, you'll see that these regimens require many hours of wake time fasting, which are problematic for hunger and mood changes. In a couple of head-to-head studies, they don't end up causing more weight loss compared to standard calorie restriction type dieting. And people don't stay on these diets for long periods of time because of this difficulty of doing the fasting during the day. One mechanism I wanted to highlight here, because it's true for most fasting regimens, is and other types of weight loss regimens, including just calorie restriction and exercise, and even metformin, which I'm not going to talk about after this slide, is that they activate AMP kinase. So AMP kinase is activated when ATP levels are low, and so many of these energetics-focused means of reducing cancer risk are going to activate AMP kinase, and fasting does this also. So in the center, you have a model of a tumor cell, and you have AMP kinase is the black oval there. So AMP kinase is directly inhibiting tumor growth by activating by inhibiting mTORC, and then indirectly it's activating inhibiting tumor growth by its activation in the peripheral metabolic tissues like the liver on the left or the skeletal muscle on the right. So increasing glucose uptake in the skeletal muscle, decreasing glucose output in the liver, all of which will reduce insulin exposure in people that are activating their AMP kinase by any of these means. So fasting is definitely activating AMP kinase as one of its cancer inhibitory mechanisms. So what about alignment of food intake with the circadian clock, aka time-restricted feeding or reducing food intake at night? A lot of different ways of doing this. So we live in a 24/7 world, a lot of us, some in the upper left corner, sitting at your desk at night, writing in your grant for the PQ, your proposal. This is a common scene, or late at night eating on the upper right in the restaurants. This is the life that we live. So this is our situation. So how does this affect us? So we know, as was mentioned, that circadian rhythms are entrained by light and food, activity, and sleep. For the context of this talk, of course, we're focusing mostly on the food entrainment of not only the peripheral tissues, the liver, metabolically active tissues, liver, heart, muscle, fat, etc., also tissues in the brain. But it's important to have these alignment of the light and the food and activity as well, but we're not going to go there for this talk, in alignment. So the food intake, alignment with the light exposure in humans who are diurnal, we are active during the day. This is the ideal alignment for humans. And disruption of this alignment is associated, on the left you'll see the list, topping the list just because I ordered it this way, with a variety of cancers and other metabolic and cognitive disorders. So what happens when people eat, or mice eat, outside of their circadian rhythm? Well, that behavior is associated with increased risk for chronic diseases and cancer. So we know that shift workers have flight attendants, people who live kind of that 24/7 lifestyle have increased risk for chronic diseases, cardiometabolic diseases, and cancer. People who are eating at night and snacking at night, there have been studies shown in large populations that these folks have increased incident breast cancer and prostate cancer. Very interesting mouse model studies of jet lag and daytime eating. They're eating out of their sleep cycle, eating during their sleep cycle is associated with increased lung cancer, liver cancer, So many models even an increased UVB-induced DNA damage in the skin. So there's a lot of very intriguing evidence here about the importance of alignment of circadian rhythms and cancer risk. Even weight loss is enhanced if the largest meal is occurring during the daytime versus in the evening. So these are two studies, a post-bariatric surgery study and a calorie restriction study from Spain. People always ask me, what about people in Spain who eat at night? So these weight loss studies were done in Spain, so I encourage you to check those out. All right. This is just a schematic of kind of activities, biochemical and physiological activities that are kind of happening at or peaking at different times of the day. And I'll draw your attention to two things that are happening in the early to mid, well, I'd say early evening. So the first star is that growth hormone peak surges are starting at about 8 or 9 o'clock at night, and melatonin secretion is also starting around this time, 9 or 10 o'clock at night. So I highlight these because these hormones are interacting negatively against the hormone insulin, which I mentioned before. So so it's important to think about what's happening in your body at different times of day. So this cartoon is very idealized, and I'll tell you why in a minute, but the red shows the secretion of insulin if a person was eating 3 meals a day, which we know that they don't, and if they stopped eating in the night, which we know that they don't. So let's just pretend that they do. But this is the idealized, that people are having their insulin secretions at night and in the day, and the melatonin secretions at night. And this is a good thing. You want to keep these hormones separated because they actually antagonize each other. But we know people are eating at night, so there is secretion at night. And in fact, people that have insulin resistance, if you look at the bottom, people that have insulin resistance are secreting even higher insulin. Remember, their postprandial levels are higher. So they're antagonizing melatonin even more so than somebody who does not have insulin resistance. And further, melatonin has anti-neoplastic activity. So if you're secreting insulin at night, you're suppressing melatonin, so possibly impacting sleep, but also interfering with melatonin's anti-tumor effects. We know that people become more, and mice become more insulin resistant, and glycemic regulation becomes more deficient throughout the day. And this study by Van Cauter is really excellent. It shows that glycemic regulation of 3 of the exact same meals consumed at different times of day on the x-axis at the bottom shows a 24-hour clock time. We'll just focus on the top graph. So this is the glucose level in the blood after 3 meals, and you can see that the area under the curve of after each meal is greater throughout the day. So this demonstrates how when we eat after the dark, in the evening time, how not only the glucose exposure is higher, but the insulin secretion, not shown here but studied, is greater. And so that's driving tumors. I'm going to just skip over this one. I have said everything here, so if you can look at this later. But circadian misalignment is driving cancer-related metabolic consequences, misalignment of food intake. Okay, here we go. So what about disruption of circadian clocks, and how does that affect cancer? So we know that tumor suppression is controlled by circadian clock, and we also know that disruption of the circadian clock promotes tumor growth. So what about time-restricted feeding in breast cancer? So Dr. Panda mentioned a study of ours that we had done in postmenopausal mice, most postmenopausal female mice. We were the first to do this model. Now also Julie Pendergast at the University of Kentucky has done this study, studies on postmenopausal females. Interestingly, by the way, she's shown that estrogen protects female mice from high-fat diet disruption of their circadian clocks, but we decided not to deal with estrogen at all and look at in these mice. So we initially showed that time-restricted feeding does work to improve metabolic outcomes in obese postmenopausal mice. So we were able to, and the leadership of Nicholas Webster at UCSD, get an R01 to study the breast cancer effects of time-restricted feeding. And we have an OBX model and also the VCD model. We fatten up the mice first for 10 weeks on the high-fat diet, and then we start the time-restricted feeding for another 10 weeks, and then we inject the tumor cells. Sorry, we do the time-restricted feeding for 4 weeks and then do the tumor cell injection. There are two other TRF models in mice that have been published so far showing reduced tumor growth with time-restricted feeding in breast and lung. There are some small studies of time-restricted feeding, including Dr. Panda's, which are very exciting to see. We need properly powered studies in humans. So I just want to go over 3 slides of our large epidemiological studies that we've done. These are important to show generalizability of these effects of nightly fasting or reducing intake at night, food intake at night. This is an Entrained study, 2 outcomes led by Ruth Patterson and Catherine Marinac, who's now at Harvard. 2,500 women in Entrained, looking at 24-hour food recall, showing that increased nighttime fasting duration was associated with lower hemoglobin A1C and also reducing with less nighttime eating, and nightly fasting was associated with lower C-reactive protein. A recent study that we have just published, we are getting ready to publish, but have presented at the American Heart Association meeting, led by Noor Makarem at Columbian University Medical College, in 13,000 Hispanic adults, men and women, that eating a greater percentage of kilocalories after 6 p.m. was associated with higher glucose, higher insulin, and insulin resistance. And most exciting for this group, that fasting for greater than 13 hours per night was associated with greater breast cancer, was associated with a reduced risk of breast cancer. So fasting less than 13 hours at night, I'm feeling rushed, less than 13 hours a night is associated with 36% increased risk of cancer recurrence. And to our knowledge, this is still the first human study to demonstrate a clinical outcome with nightly fasting. We have done a pilot study of feasibility in obese postmenopausal women. They like it, it works. And I'm going to skip over this mechanisms one. And I want to focus just on this 4th point here, that fully powered RCTs really are needed to appropriately test these interventions that utilize fasting or time-restricted food intake. And lastly, these are my collaborators and funding.
All right, thank you, Dr. Sears. Now we'll move on to our final speaker, Dr. Peterson. Dr. Courtney Peterson is an assistant professor in the Department of Nutrition Sciences at the University of Alabama, Birmingham. She received her PhD in physics from Harvard University before changing disciplines and pursuing an obesity postdoctoral fellowship in nutrition and metabolism at Pennington Biomedical Research Center in Louisiana. Dr. Peterson's research seeks to develop and test dietary strategies to mitigate or reverse diabetes and obesity, including the role of meal timing in relation to circadian rhythms and intermittent fasting.
Great. So I'm going to talk to you today about human intervention studies on intermittent fasting and time-restricted eating. So intermittent fasting is defined as any sort of meal timing intervention or approach that alternates periods of eating and extended fasting. Now, there is no one single quantitative definition that everyone agrees on. So among sort of the purists who first studied intermittent fasting, they'll often say it's anytime you fast for at least 24 hours at a time. More recently, coming out of the chronobiology community, there's been a push or a consensus more of around 12 hours a day. And then I think there's the complicating factor that the minimum duration of hours of fasting probably depends on the species. For humans, I think something closer to maybe the 14 to 18 hour range may be a better target for 2 reasons. So one, data from NHANES, the largest nutritional assessment, of individuals in the U.S. suggests that the median American eats over a 12-hour period. So I'm reluctant to say the median American is practicing intermittent fasting. And then second, if you look at what happens in terms of substrate oxidation pathways, oftentimes glycogen breakdown is still predominant around 12 hours of fasting, so a slightly longer definition may be appropriate in humans. So I'm going to cover or talk about 6 different types of intermittent fasting that have been studied from an intervention perspective in humans. And I like to group them into 3 different categories to make sense of them so you can see the commonalities. So the first type of approach I'll call periodic fasting. These are approaches where you have full 24-hour or longer fasts, some number of times per week or per month. And the classic example there is alternate day fasting, where you have a 24-hour fast every other day. Another example would be fasting for 24 hours once a week, and I won't cover that here today. The second type of approaches are what is now called intermittent energy restriction, or IER. These are approaches where you eat a very low-calorie diet some number of times per week or per month. Now, there's a little bit of debate about what these approaches do constitute intermittent fasting or not, but I would argue at least some of them do, because some of them at least involve an extension of the fasting interval between meals. And I'll talk about 4 different approaches here. And then the last set of approaches are your daily intermittent fasting approaches, which are now largely known as time-restricted feeding, which involve eating in a shorter time period each day. And as has been discussed in the previous talks, this can be achieved without necessarily reducing calorie intake, whereas I'd like to make a comparison with these other approaches that have a specific caloric target that participants are trying to meet on fasting and feasting days. So to my knowledge, one of the first trials on intermittent fasting in humans was this 2005 study that was published on alternate day fasting, which involves those full 24-hour fasts every other day. And in this study, which is about a 3-week study in 16 people, they found that alternate day fasting induces weight loss and fat loss in a zigzag-like pattern. However, it also increased hunger quite dramatically with no signs of lessening over time, suggesting that ADF may not be sustainable. So there was actually very little research in humans on ADF for more than a decade. There were no trials. More recently, we've had 2 randomized controlled trials comparing alternate day fasting to continuous energy restriction. And although they both reported great adherence, they reported conflicting results for whether ADF improves weight loss and blood pressure. The second type of approach, and the first type of approach that falls under this intermittent energy restriction category, is alternate day modified fasting, or ADMF. This is an approach that alternates fasting days, where typically participants will consume something like 25% of their normal daily caloric intake, with feast days, which are usually conceived of as ad libitum days, or where participants consume about 125% of their daily caloric needs. And this approach has been pioneered by Dr. Krista Vardy, and she's done multiple studies in rodents and humans. And to date, there are about 7 moderate terms, so about 8 to 12-week studies on ADMF in humans. And they find, by and large, that it reduces body weight, body fat percentage, visceral fat, and improves insulin, blood pressure, lipids, inflammatory markers, and adipokines. However, nearly all of these studies are single-arm studies. More recently, we got our first large-scale and long-term ADMF trial in humans. This is a 1-year-long trial in 100 participants, and the participants were randomized to a control group, to a continuous energy restriction group, which you can think of as your daily dieting approach, and then an alternate day modified fasting approach. And the last 2 groups were counseled to reduce their calories by the same net amount when averaged over the week. And unfortunately, what they found is that alternate day modified fasting is no better than continuous energy restriction at every time point tested. Moreover, there were no differences in glucose, insulin, blood pressure, heart rate, lipids, visceral fat, or C-reactive protein. And another smaller study, and that was also shorter, similarly concluded that ADMF was not better than continuous energy restriction. So then you might think, well, maybe the 2 are equivalent, so ADMF could be an alternative. However, the one caveat is there was a higher dropout rate for alternate day modified fasting. suggesting that it's actually less sustainable than conventional dieting. The one silver lining is a post-hoc analysis indicated that ADMF improves insulin resistance more than continuous energy restriction in individuals who are insulin resistant at baseline. The third type of approach, which is also an intermittent energy restriction type approach, is the 5-2 diet. This approach is hugely popular in the UK and Europe, and we don't hear about it quite as much here. But this is an approach that involves eating a very low-calorie diet 2 days a week, and those days can be either consecutive or non-consecutive. And this approach has really been pioneered by Drs. Michelle Harvey and Anthony Howell. They've completed 3 clinical trials. And then in literally the last 2 years, we've had an explosion of interest in this field, mostly out of Europe, and we've had about a dozen new trials come out. So Drs. Harvey and Howell's seminal trial is this 2013 trial, and it was a 3-month trial in over 100 overweight women with a family history of cancer. And this was one of only really 2 trials that really truly has a cancer focus. As you'll see throughout the presentation, nearly all the data we have on intermittent fasting comes from a cardiometabolic angle. Nonetheless, in this trial, they found that the 5-2 diet reduced body fat levels, and insulin resistance more than continuous energy restriction. So this was really one of our first longer-term clinical trials in humans, suggesting that intermittent fasting may have benefits, even relative to continuous energy restriction. However, they found no difference in most other fasting cardiometabolic endpoints or in the cancer marker IGF-1. Now there have been about 4 one-year-long RCTs with large sample sizes on the 5-2 diet. And interestingly, they conclude that there's no difference in weight loss success after about a year. However, 2 of the trials did find marginal improvements in weight loss at 12 weeks or at about the 3-month mark. So this suggests that maybe in the short term, these types of approaches may enhance weight loss success. However, there was no change in fasting cardiometabolic risk factors. In the one and only study that looked at postprandial values, however, they did find in that study that the 5-2 diet improved postprandial C-peptide levels and triglyceride levels more than conventional daily dieting. And unfortunately, the big problem is in nearly all of our intermittent fasting studies, we only look at fasting markers, which can actually lead to psoriasis, and I'll show you an example of that later. So we really, really need to be looking at the postprandial level as well. There have been a handful of meta-analyses now comparing intermittent energy restriction approaches to continuous energy restriction approaches for weight loss. And here's an example of one of those here. This is a meta-analysis with 11 RCTs, longer than a month, and they found that intermittent energy restriction does not enhance weight loss relative to conventional dieting. However, it does reduce insulin levels more than conventional dieting and leads to marginal improvements in HDL and triglyceride levels. This is really our best trial on any form of intermittent fasting in cancer in humans, so you can see why we so badly need this RFA. So this was actually a single-arm trial in 23 overweight women at high risk for breast cancer. And participants followed the 5-2 diet for only 1 month, and they did achieve 5% weight loss within that month and improvements in insulin sensitivity. However, when they looked at the gene expression within breast tissue biopsies, they found that only about 55% of women had positive responses to the diet. And this was a much more heterogeneous response. So I think the investigators were kind of scratching their head why more women didn't seem to respond positively to the intervention. So it's possible that maybe responses to intermittent fasting are incredibly heterogeneous, and we just don't know that yet. The fourth type of approach I want to mention, which really has more of its roots in cancer research, is the fasting-mimicking diet, which has been pioneered by Dr. Walter Longo. The fasting-mimicking diet, or FMD, involves eating a very low-calorie, low-protein, plant-based diet for several consecutive days at a time. So typically about 5 days. And then you repeat this cycle some number of times per year, but no more often than typically 1 month. And Walter came up with this sort of approach because he was doing extended fasting protocols, and he found that a very small minority of people were willing to do, say, a 48-hour fast. And he had gotten fantastic data in preclinical models showing that combining a 2-day fast with chemotherapy really led to synergistic improvements in cancer-related outcomes. So Walter's been working on this diet for many years, and in rodents, he's found that it reduces body weight, extends lifespan, reduces neoplasms. So a lot of this improvement in survival is actually driven by a reduction in neoplasms, reverses type 1 and type 2 diabetes, increases stem cell number, and regenerates organs. So to date, there have been about 3 published studies on the fasting-mimicking diet in humans and only 1 large-scale trial. So this trial, I have the data shown here, was done in about 100 healthy adults. They completed 3 cycles, or in other words, 3 months of 5 days of the fasting-mimicking diet versus a control diet. And the control diet here was weight stable, so not a conventional dieting control. And what they found is that the fasting-mimicking diet reduces body weight, body fat levels, IGF-1 levels, and blood pressure, but did not affect fasting glucose or lipids. And to my knowledge, there are now a handful or 2 clinical trials underway in Europe using the fasting-mimicking diet in cancer trials. So the 5th type of approach is known as weekly intermittent energy restriction. These are approaches where you eat a very low-calorie diet for a week or longer, and then you alternate this with periods when you're actually eating to maintain your weight. And the theory is that by this sort of alternating between caloric restriction and not caloric restriction, that actually prevents some of the metabolic slowdown we see with weight loss. So there have been a handful of trials lasting 12 weeks or longer, but the data so far seem to favor no improvement in weight loss. And the 6th type of approach are your daily intermittent fasting approaches, or time-restricted feeding in animals, or time-restricted eating in humans. So to date, there have been about 17 pilot studies in humans using 4 to 11-hour eating windows. Now, it's really important to note that every single one of these trials is either a small pilot study or is a single-arm study. Nonetheless, when you look at the body of data, when food intake is limited to early in the day, which I'm calling early time-restricted feeding, or the middle of the day, which I'm calling midday time-restricted feeding, every single one of those small clinical trials reports some sort of improvement in cardiometabolic health. Now, they don't always all agree on what endpoints are improved, but they at least agree there's some sort of benefit. And those benefits include appetite and/or food intake, weight and/or fat mass, improvements in glucose, insulin, and insulin sensitivity, fat oxidation, or what you can think of as fat burning, blood pressure, improvements in lipid profiles, oxidative stress, functional endurance capacity, and even gut microbial richness. And here's probably data from the best controlled longer-term time-restricted eating trial we have. It's actually not an RCT. They used historical controls, and they had in the intervention group, they had 23 overweight adults who followed time-restricted feeding by eating within an 8-hour window in the middle of the day. And they found after only 3 months, they reported about 2.5% weight loss, an 8-point decrease in blood pressure, but actually no improvement in other aspects of cardiometabolic disease. And participants were adherent about 5.5 days a week in this trial. Conversely, we've had 2 studies so far that have looked at what I would like to call late time-restricted feeding. And by late time-restricted feeding, I mean something that's equivalent to skipping both breakfast and lunch. So that would be equivalent to eating only dinner. And interestingly, in these 2 trials, late time-restricted feeding produced either null or negative results. For instance, in one of these trials, eating between only 5 and 9 p.m. at night worsened postprandial glucose, insulin, blood pressure, and lipid levels. And here's the data on glucose levels. And you can see if you had only measured fasting values, you would have missed the worsening in glycemic control if you had not measured at the postprandial level. So I saw some of this data on time-restricted feeding at a Sachin Panda's lab, which is fantastic, and I heard about some of Mark Mattson's early work on intermittent fasting. They had a daily intermittent fasting trial, which we might think of as equivalent to late TRF in 2008, as well as some of the work out of Oren Froy's lab looking at the old adage of eating breakfast like a king. And I thought it would be fantastic if we combine time-restricted feeding with eating earlier in the day, as opposed to many trials that have looked in the middle of the day or even later in the day. So I call early time restricted feeding equivalent to eating dinner in the afternoon. And I do think these distinctions are important because there may be some differences in the benefits depending on the time of day that time restricted feeding is practiced. So my lab has published data from 2 crossover randomized controlled trials. Both of these trials were isocaloric and eucaloric. And what that means is we provided all food to participants. We made sure that they ate it, so they were eating the food under supervision, and we matched food intake to the control group as well as made sure participants did not lose weight. And in both studies, we had an early time restricted feeding group that ate over a 6-hour period early in the day, and we compared that to a control group that ate over a 12-hour period. And what we found in our first 5-week study, which was in men with prediabetes, we found that early time restricted feeding did not affect glucose levels in the morning. So I will show you data in just a bit where we find improvements in 24-hour glucose levels. However, we did find an improvement in insulin levels in the morning. This is quite a large improvement. And it looked like as well when you look at indices of insulin resistance, that insulin sensitivity was dramatically improved as well as the responsiveness of the beta cells. We also found dramatic improvements in blood pressure, so about 10 to 11 point drop in blood pressure, and this is pretty remarkable for a clinical trial with no weight loss. We also found a reduction in oxidative stress levels. We found no changes in cholesterol levels with the exception of a large relative increase in triglycerides, but we're now thinking that may be due in fact, to free glycerol levels, which are indistinguishable from bound glycerol levels in the standard assay. In our second published trial, we did a 4-day crossover study in 11 overweight adults, and we were interested in how intermittent fasting kind of broadly speaking affects calories in versus calories out. In other words, are the benefits for weight loss due to an increase in calories burned or to eating less? And what we found is that early time restricted feeding does not affect energy expenditure. Any increases in calories burned during the daytime were offset by a decrease at nighttime. We did find improvements in the hunger hormone ghrelin, which was reduced, and we found some improvements in aspects of subjective appetite. And this, by and large, suggests that probably the weight loss effects of intermittent fasting are due solely on the food intake side, not on the energy expenditure side. We did also find an increase in fat oxidation, so fat burning was elevated for nearly a 12-hour period at nighttime. We also found in this study that early time restricted feeding lowers 24-hour glucose levels, as you can see from the 2 curves here. And not only did it lower mean values, but we also found a reduction in glycemic excursions, or what you can think of as spikes as glucose levels. And then the last thing I wanted to show you a little bit of suggestive mechanistic data we have in humans. So we found that only 4 days of early time restricted feeding tended to modestly increase ketone levels. We found not quite significant reductions in IGF-1 levels, so the p-values here are both 0.11. We found a tendency towards an increase in BDNF levels, alterations in cortisol levels, and then within blood cells, we found an increase in SIRT1, which is an anti-aging gene, an increase in one of the major autophagy genes, and widespread changes in circadian clock genes. So in addition to the 2 published studies I showed you, we have 3 larger clinical trials in progress with large sample sizes, and we're asking questions such as, does time-restricted eating enhance weight loss relative to continuous energy restriction? Does the time of day that you practice time-restricted feeding matter? And can time-restricted feeding be used as a treatment for type 2 diabetes? And to summarize, I'd say a majority of intervention studies published so far report no or a small number of differences between intermittent fasting and continuous energy restriction, at least when participants are counseled to eat the same. So to me, I think the take-home message is a lot of the benefits of different intermittent fasting approaches may just simply be due to the reduction in calories. However, some forms of intermittent fasting may have health benefits that are independent of energy restriction. And this is particularly true for the data I showed you on time-restricted eating out of our lab. I think one of the other take-home messages is that we shouldn't group all intermittent fasting regimes together. Some may be harder or easier to adhere to than conventional energy restrictions, so we really need to keep that in mind. And I'd say only a couple interventional studies have really been performed on cancer-related endpoints and mechanisms, so hence we really need a lot more studies in this area. And in general, we need more research on the efficacy, long-term effectiveness, mechanisms and feasibility of different approaches. And lastly, I'd just like to close by thanking our funders as well as the people who made this happen. And I'd like to suggest 2 review articles for future reading on intermittent fasting and cancer. Thank you.
Thank you, Dr. Peterson, and thank you again to Dr. Panda and to Dr. Sears for your excellent presentations today. So now Dr. Ed Sauter is going to introduce some more information on NCI's provocative question number 2, which is related to intermittent fasting and related approaches. Then he'll open up the floor to questions from the audience, which will allow the webinar participants to interact.
So as you've heard from our 3 speakers, the data on intermittent fasting is quite compelling, but that's primarily in animals. Despite a dramatic increase in the use of intermittent fasting among humans, we really don't understand whether these animal studies and the findings in animals really translates to humans. And the human studies, for the most part, are small in number and short in duration. And of course, one of the issues related to fasting is adherence to the fasting or the diet for a long term, because obviously if you're not going to adhere, it's not going to be effective. And so especially in the human studies, many of those studies focused on non-cancer endpoints. And so I think there really is a need to learn more about the role of intermittent fasting and its effect on cancer. And so that's what this provocative question is trying to address. So we're going to move now to our questions. The first one is a question for all 3 of our speakers, and I would like to first for Dr. Panda, if you could address this question. So there are still positive metabolic effects, at least in some of our human studies, even though the individuals did not lose weight. Why do you think that that was the case?
Well, in our mouse studies where we have looked at many gene expression and other stuff, we have seen significant improvement, for example, in AMP kinase pathway, mTOR pathways, PPAC gamma, mitochondrial response, etc., in mice that did not lose weight. I must point out that in many of our studies, we do have controlled mice which are on normal diet. They do time-restricted feeding where we do not see weight loss, but the muscle mass goes up and fat mass goes down, and we do see similar molecular changes that happen. So I think there is a overemphasis on linking obesity to cancer, and we always think that anything that reduces cancer risk or improves metabolic health has to go through weight loss, which is not true, because we got to keep in mind that nearly 1/3 of metabolic disease patients are not obese. So we have to keep that in mind, that not all benefits have to go through weight loss.
Okay, thank you. How about Dr. Sears? What are your thoughts about these observations?
Yeah, I would say that I would remind everybody that the mouse studies, which I've been doing both, doing in mouse studies and then also working in the human population studies, so thinking about this in a very translational way. The mouse studies are very short term. And so in the very short term, you see dramatic changes in metabolism, reducing fasting insulin levels, the fat in the liver just melts away so quickly. And we do see some reductions in adipose tissue in the mice. Their total body weight reduction is minimal, but these are short term. So these are, 8 weeks, 10 weeks. So I think in a human, when you extend that to a 6-month intervention, a 1-year intervention, something like that eventually I think the weight might come down. But importantly, it's these mechanistic drivers of cancer that are changing, right away, relatively quickly in the mice. And I think that we're likely to see that in the human studies as well, and that's the important thing. And I agree with Dr. Panda that we've got to kind of make a disconnect between the goal of making the number on the scale smaller and focus on the metabolic mechanistic changes.
Thank you. So thanks for mentioning that, because again, I wanted to emphasize for our provocative question, we want the folks to be on human studies. There can be mouse animal studies, but only if it's an adjunct to the human study. And the studies, if it's a clinical trial, should be at least 6 months because we want some longer-term data. So finally, Doctor, regarding this same question, Dr. Peterson, what are your thoughts?
Yeah, I think it's quite interesting. I often approach a question like that from two lenses, one of both circadian biology and the other from metabolic research, which is a little bit more from where I derive my training. There have been some really elegant studies, for instance, out of Oren Freud's lab in Israel, where they did a 1-week trial that was in weight maintenance, so there was no weight loss, where they simply changed the distribution of calories throughout the day. So one arm had participants eating most of their calories at breakfast and the other at dinner time, and they find dramatic improvements in insulin secretion and insulin sensitivity in only a week's time. And I think, one of the clear mechanisms that's independent of weight loss could just be better timing of peripheral organs, better circadian timing of peripheral organs. I think there are other mechanisms at play because, for instance, if you take blood pressure, we found very compelling results in our study that blood pressure was dramatically reduced with no weight loss in only 5 weeks' time. But they're both circadian and intermittent fasting mechanisms. So, for instance, we know that natriuresis is more efficient at nighttime, at least in animals' nighttime, at least when food intake is earlier in the activity period. So there's definitely a circadian mechanism for blood pressure. But then we also know that keeping insulin levels low also promotes a decrease in blood pressure, and there's a direct mechanistic link there. So there are a bunch of mechanisms that I think may explain, physiologic mechanisms that may explain these differences. But I think it's quite interesting because some of the best data we have comparing intermittent fasting to like a no weight loss control come from time-restricted feeding rather than other types of intermittent fasting. So time-restricted feeding may be a little bit more powerful because it also has potentially that circadian angle.
Okay, thank you. So now we're going to go to individual questions. And just a follow-up to what you were saying, Dr. Peterson. So if you're not losing weight, and adherence is so important, how would you, and I'm saying you, I'm going to ask Dr. Sears, how would you convince, as I understand it, the studies so far in humans, the adherence has not really been better in intermittent fasting studies than it has been in standard caloric restriction studies. How would you convince people on those studies and or in real life to improve adherence long term? So Dr. Sears, what's your thoughts?
So, and we did 2 pilots of 10 women each, so a total of 20 women of time-restricted feeding. Basically, we call it prolonged nightly fasting. We're focusing on the fasting period at night. And our women were compliant more than 95% of the nights that we did. So I think it's intermittent fasting where people are fasting during the day where there's a compliance issue. Fasting during the night, I think people, and this is Courtney Peterson's, Dr. Peterson and Dr. Panda's experience as well in their clinical trials, that the compliance is good if they're fasting during the night. And then I would say what we heard from the women in our pilot was that they were not hungry at night. Some of them did lose weight, and they loved that. They loved that they reported sleeping better. I mean, just some qualitative things is what I have to report about that. Maybe Dr. Peterson and Panda could share other things that would encourage people to keep on with their compliance.
Well, Dr. Panda, any thoughts?
Yeah, so we actually extensively ask our participants, and also many of our participants continue up to 1 year after the intervention, 3 months intervention has finished. The reasons why they continue, the number one reason they say is improved sleep. A lot of people sleep much better, and now that has been tested in animal models, and we don't understand why they get into deeper sleep. If they sleep better, they have much better energy during daytime, so they like that. And then another thing that we did not think about was many participants report reduced heartburn or acid reflux. We must keep in mind that in the US, there are 64 million prescriptions written every year for heartburn. Plus, there are over-the-counter medications. So when people feel good about their gut, sleep, and mood, then they get into this positive feedback loop to maintain adherence. They're not stepping on this scale every day to keep adherence. When they wake up rested in the morning, that's what drives them, in our experience.
Okay. I'm going to direct a new question to Dr. Peterson. So there's a lot of literature on the importance of family meals in promoting many aspects of well-being in youth. Is this feasible using time-restricted feeding or another intermittent fasting approach for the rest of the family?
Yeah, I think the answer is we don't really know yet. Eating is a very social phenomenon for a lot of people. My suspicion is that middle of the day, time-restricted feeding approaches, for instance, are going to be more doable for a lot of people. But it may not be that time-restricted feeding is going to work for everyone, if we're honest, because some people have, they're doing shift work or there's a specific time of day they need to eat with their family. We have a little bit of exit interview data from our studies, and it does suggest that people who are preparing meals for their family are sort of the ones who have the greatest challenges. But I think the answer is we don't have a clear sense yet. I think the other argument you can make is in all these time-restricted feeding studies, it seems like whatever improvement you have in that direction of eating in a shorter period of time, there are improvements. So in individuals, like Sachin's studies have elegantly shown, in individuals who start out with 15-hour eating periods, if you get them down to 11 hours, they're still getting a lot of benefits.
Okay, this is for Dr. Sears. Do you recommend intermittent fasting to a normal weight, healthy individual?
I would.
What would you tell them?
What I would tell them is that I would refrain from eating large meals in the nighttime after 6 p.m. based on our epidemiological studies, some of which were not, solely done on, our NHANES work was not done on obese women. It was all on women, but we had no BMI cutoff. And so consuming less than 30% of one's calories during that period of time seems to be advisable based on our work in those population studies.
Okay, next question. Thank you. This is to Dr. Panda. What do you think is the importance of mood and hunger in sustainability of these intermittent fasting regimens?
Yeah, as I pointed out, mood and hunger are very important. And in our first paper in 2015, in our exit interview, we found that people who did 10 or 11 hours of time-restricted eating had less hunger at bedtime. And they also slept better. So when they sleep better, mood improves, and that helps them to stick to time-restricted eating.
Thank you. So, Dr. Peterson, what do you think is the importance of intermittent fasting and specifically time-restricted eating in response to cancer therapy, for example, chemotherapy?
Yeah, in human studies, we have no data. In animal studies, Walter Longo, for instance, has some really elegant data comparing fasting for, I think it's 24 hours, 48 hours, 72 hours. And he finds, this is quite interesting, and I'm glad you brought it up, he finds the sweet spot of 48 hours of fasting initiated prior to chemotherapy produces the best outcomes in terms of killing the tumorous cells. If you initiate the fasting after the introduction of chemotherapy, you don't get the same synergistic outcomes. So that's some of the most interesting data. I think he's called this a differential stress response. And there's data that suggests that both the healthier cells are more resistant to the chemotherapy, as well as the tumor cells are rendered more susceptible to the chemotherapy. And as far as I know, there's very, very little data on immunotherapy. So we have a lot of work to do in this area. There was a bunch of research actually in the 1970s trying to look at the administration of cancer drugs, the timing throughout the day. So you could think of that as the circadian timing of drugs, and that may also play a factor, though some of the data on that's been conflicting. So I think there's great potential there for the application of intermittent fasting or even other circadian modalities.
Okay, so Dr. Sears, how can morning, daytime, or evening physical activity have an effect on insulin or melatonin?
So exercise is not my strength, but my readings have shown that exercise in the morning tends to be better, but I think actually Dr. Panda would have a better grasp on this literature. Dr. Panda?
Yeah, so this is a new exploding area. There is some historical data correlating better exercise capacity and reduced risk of injury if people exercise late afternoon, say up to 2 hours before bedtime. The new studies are finding that that's true, and it has some interesting link to glucose regulation. Type 2 diabetics who exercise in the morning, and this is a specific type of exercise, high-intensity interval training, they may or may not improve their glucose. And in fact, it can worsen their continuous glucose, 24 hours glucose, whereas the same individuals, when they crossed over to evening exercise and identical exercise, then they improved their glucose regulation, their 24 hours glucose and morning fasting glucose reduced. This came out of Stockholm early last year. We are yet to see whether it's reproduced in other studies and whether the high-intensity interval training is the only exercise that has this differential glucose response. By the end of the day, I guess exercise is a good medicine. Do it. And then if people have to optimize, then they have to pay attention to time.
And I'll insert one more comment, which is if your main outcome is not exercise performance, like Dr. Panda mentioned, but is instead weight loss and fat loss, there are now at least 2 studies showing that exercising fasting in the morning does enhance or improve weight loss and fat loss. So, there's a little bit of difference depending on the time of day and what you're going for.
Thank you.
Thank you.
All right, thank you everyone for an excellent presentation today. I know we had a lot of great questions come in, but I want to be respectful of everyone's time, and we are coming up on 2:30. So thank you everyone for your participation in today's webinar. As I mentioned earlier at the very beginning of the webinar, this presentation will be posted online in the next couple of weeks. And please feel free to continue the conversation via email if you have any additional questions or maybe any questions that were not addressed during today's presentation. So thank you everyone for your time and have a wonderful day.
Thank you.